Okay, let's move to chapter 9. Oh, let me put the microphone on. Chapter 9, patterns of inheritance. So we talk about genetics. Which is a big topic. Okay, why genetics matter? Well, we inherit everything we get from a trace from our parents, and it's really important that, to know really something about that. You know, your sex was entirely determined by your father, and remember why? Because eggs come in one variety with an X, sperm come in two varieties, X or Y. And if the X sperm fertilizes the egg, you have a little baby girl. The Y sperm, you have a little baby boy. So that determines the gender of the baby in human beings and any other organism that has the same kind of gender determination. Okay. Some features entirely determined solely by genes, some by environment, some by both. This is the old nature and nurture controversy. How much of us is hardwired in our genes? And how much of us is shaped by our environment and our upbringing? And your environment can actually change the expression of some of your genes. It's called epigenetics. Okay, your chance of inheriting and passing on genetic disease can also be determined from your family history. So you may have someone in your family with a genetic disease, and you can find out what is the chance that you are a carrier for this. That is, most genetic diseases are recessive, and so each one of us is actually a carrier of some kind of bad recessive gene. And so we're going to look at pedigrees later on, and how you can look at a pedigree and determine, what is your chance that you are carrying this particular bad allele. You just think of, dogs and cats and horses as having pedigrees, but so do people. Any sexually reproducing organisms have a pedigree. Okay, a good example of how genetics can really modify the look of organisms is by dog breeding. People have selected and mated dogs who prefer traits for more than 15,000 years. Thousands of years genetic tinkering has led to the incredible variety of body types that behaviors in dogs live. And this is really a form of genetic engineering. By selectively breeding, what dog do we want to breed with what other dog? And we talk about genetically modified organisms. Well, all of our dog breeds and cats and all of the animals that we bred for thousands of years to either be our companions or to eat them are genetically modified organisms. They're all. So we're all eating genetically modified organisms except for wild caught. What's the last remnant of our hunter-gatherer history? Gathering wild animals for food is commercial fishing, wild caught fish. A lot of fish now are bred also in ponds, and they're captively bred, but wild fish, and if you'll go out deer hunting or something and kill deer in the woods, you're being a hunter-gatherer. So that's the old form of genetic modification. So almost everything we eat is a GMO. But when they put on no GMOs used in this food on a food label, for example, what they mean is transgenic organisms that have had genes from one organism put into another organism. And we'll talk about some of those. We'll look at what's called BT corn, and we'll look at some fish strawberries. These are genetically modified in a new way of being able to put genes from one species into another. But genetic modification can be just selective breeding. And we've been doing that for thousands of years. Okay. So people have done this for thousands of years, but the biological principles underlying how that happened are only been recently understood for the past, actually, for the past maybe 100, 120 years. So, dogs bred from wolves, and uh, and these dogs are not different species, but look how incredibly variable they are, you know. And they're not different species because we're going to define species, uh, and that's a group of organisms that can interbreed. So, um, can any dog species interbreed with other dogs? Can any dog breed into any dog variety interbreed with the other dog variety? Yeah, you can get uh, poodles and huskies, or um, German shepherds and Basenjis. They mate. Now, can you get a Chihuahua and a Great Dane to mate? Well, you have to assist that somehow. I did hear about a Chihuahua actually inseminating a male Chihuahua inseminating a Rottweiler, but she was lying down. But they can still achieve it. Now, give us a few more thousand years, and maybe they'll have two dog breeds that we've changed so much that they can no longer actually breed. And then we've actually made, it's like artificial species. Okay, heredity is the transmission of traits from one generation to the next. And genetics is a scientific study of heredity. And it used to be just called heredity or inheritance, because people really didn't know. They knew some things about it. Well, they had to, otherwise they wouldn't have known to make all these varieties of these domestic animals that we have. They knew that something was being passed on from parents to offspring, and you could sometimes concentrate a better trait if you wanted a bigger tomato. Find one plant with big tomatoes and mate it with another plant with big tomatoes, and more likely than not, the offspring was going to have big tomatoes and even bigger tomatoes. So that's how we end up. The ancestor of the tomato had fruits about this big. Tomatoes are fruit. Now we got beef steak tomatoes that are like this big. Okay, the word genetics hasn't been used for that long because people didn't know what the inheriting things were. What was it that offspring was inheriting from their parents, and now we know them as genes. They were Mendel, which we'll talk a lot about, who figured out the basic laws of heredity. Called them factors. Now we call them genes. Okay. Well, there he is, right there. Worked in the 1860s, and found out that parents pass on to their organism discrete genes, which are called heritable factors. The genes are responsible for inherited traits, and genes retain your individual identities generation after generation after generation. So we'll talk about Gregor Mendel, who in the well, there he is. He was a monk in the late 1850s and 60s, and he discovered he figured out the basic rules of genetics, and he came up with two laws that he thought apply to all heritable traits. They don't, but they're still really important traits. And he did some meticulously laborious experiments with pea plants. And then his work was lost for about 40 years. Because he published his stuff in some obscure journal that nobody read, and he died in obscurity. But then 40 years after his death, some other people figured out the same things and looked back in the literature and found this paper and realized, oh, this Austrian monk 40 years ago figured out this same stuff. Unfortunately, they didn't try to hide it, take credit for it. They told the world about Gregor Mendel. So he'd be really surprised if he came back to life to realize that his name is mentioned in biology classrooms all over the world. So Mendel probably chose to study garden peas. Well, several reasons he chose garden peas. First of all, the abbot in his monastery didn't want the big animals being used, and didn't have that much room. But they did have a lot of garden plots, so let's use a plant. And peas are really easy to grow. I mean, even I can, I've got a black thumb, not a green one, but even I can grow peas. I mean, they're really easy to grow. And they come in very distinguishable varieties, and they have traits that are very distinguishable. Okay? But the other thing that this PowerPoint doesn't mention is that they can self. They can not only have sex for themselves, they can make babies all by themselves, and that's kind of a rare characteristic even among plants. And this would save Mendel a lot of work. All right. Yeah. So I guess this is implied by the last bullet there. Perhaps the most important advantages of pea plants as an experimental model was that Mendel could strictly control their reproduction. Right? Okay, so we got to delve into a little bit of plant sex to figure this out. Here's a flower, and flower is a plant reproductive organs. There's petals, but the important thing is the male and female parts of the plant. Now, some plants have separate male and female individuals, and that have male flowers, and this individual over here has female flowers, like red maple tree. They are in two complete genders. There's a male red maple tree over here that has male flowers, and they have stamens, which are the pollen-producing structures. And pollen is virtually plant sperm. Remember, that's what a male, how we define male. Males make sperm. And so this red maple tree makes sperm. It's male. And then there's another tree over here that has flowers that have carpels in them. And that's where eggs are produced. So that's a female tree over there. And what happens is pollen from this tree over here gets blown into the air and lands on the it's called a pistil. It's a sticky area on top of the carpel, and they get stuck on there, and they burrow down into the carpel to find the eggs, and fertilization happens there, and we make new individuals, which we make seeds. So that's two different genders. Now, a lot of plants have what are called perfect flowers. A perfect flower is like a hermaphroditic flower. It has both male and female parts. It has a carpel and it's got stamens. And for most, and there's a lot of plants like this, but for most plants that have perfect flowers, they cannot, if the pollen falls on the carpel of the same flower in the same individual, they don't make babies. So they cannot self, but peas can, and that was a really, I think, important characteristic for Mendel. Okay, so when Mendel wanted to fertilize one plant with a pollen from a different plant, he pollinated the plants by hand. So this was really meticulous work and tedious, meticulous work, and therefore was always sure of the parentages of new plants. But before Mendel could get started, he created what are called purebred varieties of plants. Now, what is a purebred variety? Well, let's look at one characteristic to begin with. He looked at 7 characteristics in pea plants. One of them was flower color. pea plants have two different colors of their flowers, white flowers, purple flowers. So what a purebred variety is, if you mate two purple flower plants together, their offspring only has purple. And since they can self, what you can do is you bag the flowers before the stamens get ready to release pollen. You bag the flowers so no pollen from the wind can land on, from another plant can land on those. And you take a purple plant and you bag all its flowers and you let it have sex with itself. And then make babies all by itself. And then you look at the offspring. And you know, this took a long time, and this was, let's say it was tedious work, it was laborious, and you had to wait a long term to get results because you bag the flowers now in the summer. Then you collect the peas in the fall. You store them over winter. You plant it in the spring, and then in late spring, you get the result, right? So you got to wait all year to get the results of your cross. So you had to be really patient. So you bred the two purple plants together, and if there was a white, and if from that, and then you looked at the babies, and if there was a white baby, you got to wait it out. And then it took two more purple flower plants from those offspring, bagged those flowers, and did it again. If there's any white ones coming from that, he threw them away. I don't know how many years he did this until he was sure that purple and purple breeds only purple offspring. Did the same thing for the white. White and white, only white offspring. So if you found a purple offspring, he would get rid of that. But here's the thing, he didn't know at the time, but he didn't have to worry about it because white was recessive. And if you're white, you can only, and if two whites make babies, they can only have white offspring. But he didn't know that yet. Okay, so we got purebred varieties of purple flower plants and white flower plants. Okay, so I'm gonna go through this technique here. Like I said, this laborious technique for cross-fertilizing pea plants. And when we say cross-fertilizing, that means now he was doing purple and purple and white and white. Now he's going to do white and purple. Breed a white flower plant with a purple flower plant, and that's called a monohybrid cross. Hybrid or cross-fertilization, breeding two parents that have the same character but different traits. Oh, I skipped over that, didn't I? A character is a heritable trait that varies among individuals. So a character would be flower color. A trait is a variant of the character. Right? So there are two traits. There's purple flowers, and there's white flowers. See? So each of the characters Mendel studied occurred in two distinct traits: tall plants and short plants. Yellow seeds versus green seeds, and so on and so forth. There's 7 of them. But we're going to look right now at flower color. Okay. So he's going to make two plants that differ in a particular, with the same character but different traits: white flowered and purple flowered. Okay. First, he has to remove the stamens from the purple flower, right? So essentially, he has to emasculate the purple flower, all right? And then he takes pollen with a brush from a white stamens on a white flower and puts them on the carpel of the purple flower plant. And then he bags it to make sure that no other pollen from the air can land on it. So he knows exactly where this pollen has come from. Okay, so then this carpel matures into a pod and forms peas. He stores the peas over winter, he plants them, he looks at the offspring. So this first generation is called the P generation, the parents. The offspring from that generation is called the F sub 1 generation. Right? So hybrids are the offspring of two different purebred varieties. The cross-fertilization itself is called a genetic cross or monohybrid cross. Hybrid means they differ in the characteristic, and mono means you're looking at one characteristic, one character. Later on, we'll look at what's called dihybrid cross. We'll look at two characters at the same time. But this is a monohybrid cross. Okay, the parental plants are the P generation. Their hybrid offspring are the F sub 1 generation. And then when you cross members of the F1 generation, you get the F2 generation. Okay, so Mendel performed many experiments in which he tracked the inheritance of characters such as flower color that occur as two alternative traits. So the results led him to formulate several hypotheses about inheritance. These are 7 traits: flower color, flower position, seed color, seed shape, pod color, and stem length. And we're going to learn that in each one of these characters, there's a dominant trait and there's a recessive trait. And we're going to define that later on. So there's a pea plant. Okay, so figure 9.5 shows a cross between a purebred pea plant with purple flowers and a purebred pea plant with white flowers. Now, we've been to figure out that the gene for white flowers did that, well, the gene, like I say, he didn't use the term gene, that the heritable factor for white flowers did not disappear in the F1 plants, but was somehow hidden or masked. Well, they're getting ahead of themselves here. You find out that there were, and people had known about this for a long time, way before Mendel, that there were certain hidden traits that would be in one generation, and then they'd not be in the next generation, maybe not even in the next generation. And then all of a sudden they pop up in a third or fourth generation. So they seem to hide for a while and then come back. So he found out that white was one of these characteristics. It was just, we find out that in the F1 generation, there were no white flowers. So the white was hidden. But in the F2, they came out again. So they were just hidden. They were still there in the purple flowers, the factor for white flower, but they just weren't coming out. So he deduced that the F1 plants must have carried 2 factors for the flower color character, one for purple and one for white. So here's what we figured out. Every a parent has 2 factors for a particular character, right? But they only give one of them to their offspring, all right? So from these results and others, Mendel developed 4 hypotheses. So here's the P generation, purebred parents, purple flowers, white flowers. F1 generation, all plants are purple flowers, 100% purple. Okay, then the F generation, then he mates them among themselves to make the F2. And this is how selfing again was really important, because he didn't have to go in there and cut off stamens and move pollen from one purple flower plant to another purple flower. He just had to bag the flowers before the pollen was made. So that now the pollen is made and hits the stamen on the same flower, the same plant, and they self. And so he saved himself a lot of work there. Okay, then in the F2 generation, what happens is he looks at the F2 generation, finds out that white came back. White's back. And here's the really sort of amazing thing about this. And it seems so obvious to us now, but back then it wasn't obvious. People knew about these hidden traits would come back. But the huge monumental step that he took, it sounds so simple now, is he looked at the ratio between the purple ones and the white ones. What percent of the F2 have purple? What percent of the F2 have white? Nobody had done this before. So he, when he first did, he got 3/4 of the plants have purple flowers, 1/4 have white, 75-25. And, but of course, this is just like one cross. So that's going to be some kind of random thing. He doesn't know whether that's the case or not. So he probably did it again. And he got the same result, 3/4 and 1/4. Now, it's not exactly 75 and 25, but it's real close, you know, like 73-27 or something. And then also, for every one of his 7 traits, he got the same ratio, 3-to-1 in the F2. So now he's thinking, okay, I got something here, because it's always coming out the same. So, so that's when he started to formulate his 2 laws. So I think they're coming up right here. Okay, there are alternative versions of genes that account for variations in inherent characters. I think I mentioned this before in an earlier lecture. So you have the same gene. You got a gene for flower color, all right? That codes for proteins that determine flower color. But there's 2 variations of these genes, and these are called alleles. That one allele is an allele for purple flowers, that produce purple flowers. One allele that fits at the same locus, does the same thing, determines flower color, but a different flower color, white. So, so these are alternative versions of the same gene. Okay, alleles. 2, for every inherent character, an organism inherits 2 alleles, 1 from each parent. So the parent has 2 alleles for flower color, but he only gives his, but there's only 1 of those alleles in the egg or the sperm. So, you get 1 allele. Everybody has 2 alleles for this character, 1 from each parent. Okay. An organism has 2 identical alleles for a gene. It's said to be homozygous for that gene, at that locus, at the gene locus. You're homozygous if both your parents give you the same allele. If you get 2 different alleles, that one parent gives you 1 allele and the other parent gives you the other allele, then you are heterozygous at that locus, at that gene locus. Okay, if an organism has 2 different alleles for a gene, 1 allele determines the organism's appearance. It's called a dominant allele. So to have the dominant trait, dominant expression of this character, you only need 1 dominant allele. So if you're so if you what's called homozygous dominant, have 2 big letters, you have the dominant expression of the trait. If you have a big letter and a small letter, and you have just 1 dominant and the other one is recessive, you still have the same expression, the dominant expression. The allele has no noticeable effect on an organism's appearance, and it's called the recessive allele. So geneticists use uppercase letters, such as big P, to represent dominant alleles, and lowercase, usually italics. Well, we don't usually use italics. And lowercase letters, such as small p, represent recessive alleles. So for the 2 alleles, and this is important to remember for Punnett squares and everything, you always use the same letter, the dominant and the recessive. You use the uppercase for the dominant, the lowercase of the same letter for the recessive. And so in order to have the recessive expression of the trait, you have to have 2 small letters. That's the only way. That's called homozygous recessive. Okay. A sperm or egg carries only 1 allele for each inherent character because the 2 alleles for characters segregate and they separate from each other during the production of gametes. So when when parents make gametes, they segregate, they separate the alleles. And in what process do they do this? Mendel didn't know what process it was, but we know it as meiosis. That's how alleles are segregated in meiosis. So the segregation of alleles, separation of alleles, he called a law of segregation. Every parent has two factors, alleles, for this character, but they separate them in the process of making gametes in meiosis, and they only give one of them to their offspring. And then when the sperm and egg when sperm and egg unite at fertilization, each contributes to the alleles, restoring the parent condition in the offspring. So now you're back to having two alleles for every character. So figure 9.6 illustrates Mendel's law of segregation, which explains the inheritance pattern shown in figure 9.5. So here we go to P generation. So purebred variety are going to be homozygous. So purebred purple flowers are going to be big P, big P. Purebred white flowers are going to be small p, small p. All right. And so big P, purebred purple can only give a big P to its offspring. White flowers can only give a small p to their offspring. So they mate, and all of the F1 generation are heterozygous. They have one big P from one parent and a small p from the other parent. Oh, by the way, he cut off the stamens of purple and then took white pollen and put it on a purple. I'm sure he did the reverse also. Cut the stamens off white and took pollen from the purple and put it on a white. And we found out that the results were exactly the same. Doesn't matter which parent gives which allele. Okay. So then the purple flower, the F1 offspring is all heterozygous. So now you have a choice. They can give their offspring a big P or a small p. And they segregate it out, and it's completely random which one they give to their offspring. So then you have to get the ratio for F2, you have to do the Punnett square. You put big, what's called the genotype, the combination of alleles at a particular locus. So sperm from one F1 plant has big P, small p. Eggs from the F1 plant is also big P, small p. So then you bring them across and drop them down, you end up with a ratio really of one homozygous dominant. So there's only four combinations. One of them is big P, homozygous dominant. 2 out of the 4 is heterozygous. 1 out of the 4 is small p, small p. But since big P is dominant, 3 out of the 4, the phenotype ratio. Remember what phenotype is, the way you look on the outside. So the phenotype ratio is 3 purple to 1. So genotype ratio, 1 big P, 2 big P, small p, 1 small p, but the phenotype ratio, 3 purple to 1 white. So this is this is just the same thing. Purebred, all the F1s are heterozygous. And then they want to figure out what the ratio is. Do the Punnett square here, and you get 3 to 1, purple, white. So a Punnett square highlights the 4 possible combinations of gametes, and it results in 4 possible offspring in the F2 generation. Each square represents an equally probable product of fertilization. So geneticists distinguish between an organism's physical appearance, its phenotype. And that physical appearance is not always noticeable from the outside, but it's still a phenotype. Because later on we'll talk about human blood types. That's a phenotype. I'm A positive. You can't tell that much by looking at it, but it's still a phenotype. Right? You can figure it out by tests, but anyway, it's still a phenotype. But the genetic makeup, which is what combination of alleles do you have at a particular gene locus, is your genotype. Right? What allele combinations do you have? So Mendel found that each of the 7 characters he studied had the same inheritance pattern. One of 2 parental traits disappeared in the F1 generation, only to reappear in the F2. So the underlying mechanism explained by Mendel's law of segregation: pairs of alleles segregate during gamete formation, and only one allele is given by each parent, and the fusion of gametes and fertilization creates the allele pairs again. So, as I said, and this happens, segregation happens in meiosis. He didn't know how they were segregated. We know now it's meiosis. Okay, diagram figure 9.7 shows a pair of homologous chromosomes, chromosomes that carry alleles of the same genes. Remember, that's what a homologous chromosome. A gene locus is a specific location of a gene among a chromosome. So alleles of a gene reside at the same locus on homologous chromosomes. However, the 2 chromosomes may be either identical alleles and be homozygous, or different ones, and then they will be heterozygous. So here is the relationship between alleles and homologous chromosomes. And here's the thing: Mendel didn't know anything about chromosomes. He didn't know chromosomes or homologous chromosomes were in it. So here's the homologous chromosome. It has the P locus here. It has the A locus here that has genes for some other character, and you get the B locus here that's responsible for some other character. They may not be completely unrelated to each other, just different characters of an organism. And so the genotype for this individual at the P locus is homozygous dominant, big P, big P. At the A locus, that codes for some other protein that responsible for some other character, they are this individual is homozygous recessive at that locus. That the B locus makes genes that for some other kind of characteristic, it happens to be heterozygous at that locus, a big P and a big B and a small b. So it has the at the P locus, it has the dominant phenotype. At the B locus, it has the dominant phenotype. But at the A locus, it has the recessive phenotype. Okay. So Mendel deduced his law of segregation by following one character through 2 generations. So as I mentioned this, a cross between 2 individuals that are heterozygous for one character is called a monohybrid cross. Mono for looking at one character, and hybrid for because the 2 parents differ for that character, differ phenotypically from that character. And then we're going to get into a dihybrid cross. A cross between 2 organisms that are each heterozygous for 2 characters being followed. And then we'll get into Mendel's second law. And I'm going to do that in the next lecture. But here's a checkpoint. If 2 plants have the same genotype, must they have the same phenotype? Well, yeah, they do. If they have the same genotype, they have the same phenotype. If they're big P, they're purple. If they're big P, small p, they're both big P, small p, they're purple. If they're both small p, they're red. But if 2 plants have the same phenotype, must they have the same genotype? That one is no. Because 2 purple plants, they could have a different genotype. Now, if they're all the white flower plants, they have the same genotype. Because the only way you can get white is 2 small letters. But for the dominant expression of the character, they could have different genotypes, because big P has a dominant expression, and big P, small p has the same dominant expression. Okay, so I hope you understand that. Okay, so on that note, let's stop. This will be 106 genetics 1. I'll catch you in the next, and we'll continue on this chapter in the next video lecture. So long.